Hello, everybody. Again, feels a bit like a radio Genomics Aotearoa, but good to see you all here. Here in Dunedin, just for the weather forecast for you, it's pouring with rain and it's cold. I hope it's better for you. And I would like to say a big thank you to all of you in Auckland who are taking the hit for the rest of the country. You guys are awesome. Stay strong. We might let you out one day. Today's seminar is one I've been looking forward to enormously because Maja is always an entertaining person. So Maja is from the Australian National University. She's studied all over the world, including Poland and Germany, University of Michigan, and the University of Queensland, which I understand still exists, but is now settled in Australian National University and is doing some remarkable work linking the genomics of sponges to both their development and their ecology and all the other cool things that sponges do. And she will be speaking today, sorry, the title's over here, she'll be presenting on sponge meta and epi-genomes as windows into animal evolution. So Maja, thank you so much for talking to us all the way from Australia, and take it away. I'll stay here while you get your slides sorted. Go,
thank you, Peter. It's a great pleasure to be invited. I would much rather be there in person, but I don't think it's ever going to happen again. I try to be more optimistic later. As I said a moment ago, it's going to be very eclectic. I'll just give you glimpses of what we are doing, and I hope that can be a start of a conversation later on. I'm going to start by telling you the most important thing, which is who did the work actually. And I'm going to present kind of eclectic work combination done on two different model species, sponge called Sycon ciliatum that we work on in Norway, and sponge called Sycon capricorn that we work on in Australia. And there's a long list of people who were involved in a variety of ways. And there is also those people that you see underlined are those names that will be appearing over and over because they actually produce the data. Now the title is a bit complex. It says sponge meta and epi-genomes as windows into animal evolution. And I should probably start by telling you that what I'm really interested in is animal evolution. So I'm not a sponge person, I'm not a genomicist, I'm sorry about telling you now, but I am interested in animal evolution, I'm interested in animal development and the combination of those two. And if you do not know what sponges are, then you can feel very comfortable with that because nobody knows what sponges are. Now this is one of my favorite quotations from Ernst Haeckel, 1870, and he said, "No other class of the animal or vegetable kingdom containing an equal number of abundant, large, and multifarious forms, has left naturalists, even up to the most recent times, so much in doubt as to its true nature, or called forth such a number of contradictory opinions." And the funny thing is that this was 1870, but nothing has changed. We are still very much in doubt of the nature of sponges, and we have a lot of contradictory opinions. And I'm going to tell you a bit about those opinions and the nature of sponges through my talk. All right, so they are definitely abundant. They are, as you probably know, all aquatic. Majority of them are marine. They live in the kelp forest and in the deep ocean and everywhere in between. They are large. Some of them could swallow a human if they were really up to that. And then some of them are really beautiful, and a lot of them have very, very different body forms. And why do we care? Why do I care, for example? They are used for 3 areas of research that are important to different people to different extent. And as I said, the one that I care most about is the insight into the evolutionary origin of animals. And this is because we think they might have retained features of our ancestors, although this is where a lot of doubt and contradictions come in. Something else that biologists for the past 100, 150 years were trying to understand is mechanism of regeneration, because sponges have really magical abilities to regenerate. And this isn't really why I set up to study sponges, but I found it more and more exciting as I was going on. And then something that I really do not study, but that is of interest to many people, is the fact that sponges are a source of biologically active metabolites. There are cancer and antiviral and antifungal drugs developed from sponges. And those have been found to often be produced not by sponges itself, but by the microbial partners that are living in close symbiosis with sponges. And I will not be talking about metabolites at all, so if that's what you wanted to hear about, I apologize, but I will be telling you a bit about sponge-bacterial symbiosis because it turns out to be pretty exciting. All right, so let's start with the first part, which is how are sponges a link between colonial protists and true animals? And this is a phylogenetic tree, which is a true tree, and by that I mean, while there is a whole forest of phylogenetic trees produced by a lot of people who have very strong opinions on which of those trees are true, this one is true because it includes only 4 branches. These are bilaterians, represented by vertebrates, this little shark here, cnidarians, which includes jellyfish, freshwater polyps such as hydra, and of course corals and sea anemones, sponges, which I represent here by my favorite sponges, which are calcareous sponges, and conoflagellates, which nobody doubts that they are the sister group to animals, and which are either single cell or colonial protists. Now, the reason why I say it's true is that it doesn't include a number of other phyla that people get very upset about, which are comb jellies, ctenophores, or placozoans. So we are just going to ignore those, but I'm of course happy to discuss those weird creatures a bit later. All right, so why do I say they are a link between colonial protists and true animals? This is because if you look at a sponge, if you look at the cells in the tissue, you will find there are 2 epithelial layers. The outer one is made of flat cells called pinacocytes, and you don't have to think about them too much now, but the inner layer is composed of collar cells or choanocytes. And those cells have a microvillar collar, and they have a flagellum that is beating, and this beating flagellum is propelling water to the sponge. There are little pores, water comes in, bacteria and microalgae and small particles are captured by the choanocytes, and then the water is expelled through the major opening called the osculum. Now, the cool thing about choanocytes is that they look very much like conoflagellates, to the level that some people insist up to this day that sponges have evolved independently from conoflagellates. But of course, there is also a kind of independent view of that, which is that this is simply a collar cell morphology. There is a beating flagellum and microvillar collar that is mainly used for increasing the surface of the cell, and this is an example of convergent evolution. Let's just ignore that for the moment. Now, on the other side of sponges, you have cnidarians with this pretty, normally pretty, but a very simplified drawing, coral polyp or a hydra. And you can think of a sponge, especially a calcareous sponge like this one, and a coral polyp or a freshwater polyp as having very similar body plans. They have the ectoderm, pinacoderm here, and true ectoderm here in the polyp, and then they have this inner layer which is responsible for absorbing and absorbing the nutrients, although in a bit different ways, which is called entoderm in cnidarians. And then on top of that, you have the apical opening, which here is the mouth and the anus, because the food comes in and comes out. And for sponges, this is an exhalant canal through which, or opening through which waste products are being thrown out. And then I will not go into much details of the homology between the endoderm or entoderm with the endoderm and mesoderm in bilaterians, but it is broadly accepted that the gut, which is of endodermal origin, and the endodermal part of the polyp are homologous. All right, so that is very kind of 19th century thinking, but we can now, because we have the genomes and we have ability to look at gene expression, we can now start testing those potential homologies. And I'm not going to talk about potential homologies tested between conoflagellates and choanocytes, simply because I don't have time for that. But we can talk during this seminar of the homology between, potential homology between the endoderm and cnidarian, between the choanoderm and the cnidarian endoderm, because there has been a lot of studies that were addressing that between cnidarians and vertebrates and other bilaterians. All right, so when I talk about developmental regulatory genes, I mean components of signaling pathways and transcription factors, and work on cnidarians and bilaterians for a very long time has demonstrated that the major body plan of all animals, including cnidarians, is patterned by very conserved set of pathways. So you will be seeing quite a few of those images. The animal is clear, and the dark purple color indicates gene expression. This is an effect of in situ hybridization, which is done the old-fashioned way, meaning we are developing color using a purple precipitating substrate. So in very simple terms, if you are not used to looking at those images, the dark purple color indicates a gene of interest was expressed there. All right, so if you are a polyp, you have the Wnt pathway expressed around your mouth. And if you are a bilaterian, you have, such as this Amphioxus lancelet embryo here, you have Wnt expression around your posterior end, which is usually confined to early stages of development. But of course, if you are a bilaterian that has that grows constantly and is able to regenerate and reproduce, regenerate and divide and so on, such as some flatworms or planarians, then you have the Wnt expression associated with your posterior pole throughout your life. All right, so the Wnt pathway is well known to be responsible for settling the main body axis, and we can generally see it by looking at the expression of the ligands of the pathway that are activating the downstream components. There is a second body axis present in bilaterians and in some cnidarians, and this is patterned by the TGF-beta and in particular the BMP pathway. And we know, for example, that our, vertebrate ventral part, the non-neural part during development, has expression of BMPs. And if you are an insect, it's exactly opposite. You have expression of BMP on your dorsal part where your nervous system is not forming. So together, the Wnt pathway and the TGF-beta pathway form this perpendicular way of two perpendicular axes that are forming the major body plan. And then the next thing that the animal has to do during its development is not only to decide what is the main axis and the secondary axis, but also how to specify the inner from the outer. And the endomesoderm specification, so specification of the cells that in later development will give rise to your gut and muscle and all those associated structures, is again specified by a very conserved set of transcription factors, including GATA and brachyuria and so on. And again, this is GATA expressed in the developing gut of a mouse embryo, and this is GATA expressed in the endomesoderm of a sea anemone nematostella. All right, so this is what people have known for years. That has been a lot of excitement about that for years. Now we accept that. But how about sponges? Because this is what I really wanted to tell you about. So when I moved to Norway in 2007, we have decided to start working on this sponge. This is called Cyclone ciliatum. It is abundant through quite a large portion of the Northern Hemisphere waters, and we have worked with the population from Norway. We have sequenced the metagenome. We'll get to the meta part a bit later, and we've done in-house assembly from using Illumina technology, which at that time was the sensible way to go. These were simple per end and made per libraries. And we ended up with, well, I think at that time, 180 kb of an N50 wasn't that shabby. And the genome size is approximately 360,000 base pairs. We have also developed extensive transcriptome and resources and hybridization protocols. So this is a bit of old work, but I would like you to know it because it's going to be important for what we have been doing next. So my postdoc Sven back in Norway was addressing the question of homology of cnidarian and sponge body plan by first identifying it was more of a group effort, and then looking at gene expression of those classical charismatic developmental regulatory genes, such as the Wnt ligands and the TGF-beta pathway components and those transcription factors such as GATA. So what we found, to our great delight, and what we think would have delight Ernst Haeckel, who quite liked this idea, or who developed this idea originally back in the 1870s, is that as in cnidarians, we have expression of Wnt associated with this major body plan opening, osculum here, such as the mouth here. The TGF-beta pathway components are all expressed radially. There is no second body axis. We never expected that, but they also appear to be associated with the osculum, with the opening. And then all those transcription factors that we could find in the genome that are associated with endomesoderm specification in other animals, these are found expressed in the choanocytes, so in the inner layer that is digestive. So what do we conclude? And I hope, I apologize for having this red and green combination here, but we conclude that our results are consistent with homology of the green layer, the endoderm here in the cnidarians, and the choanoderm in sponges. And then we also are very strongly supporting the idea that the mouth of the polyp and the osculum of sponges are homologous structures. There was also a very nice work done in Sally Lee's lab on different sponges, where they have found that if you transplant parts of the osculum to another sponge, you are going to reorganize the entire system and generate other body access. And for those of you familiar with developmental biology, that means those structures have the oscular area has the organizer capacity, which is known to be from development of animals from amphibians to cnidarians and back, and which is linked to all those signaling molecules and their antagonists. So this appears also to be conserved. So I would like to leave you with the idea that the genes that we are finding are supporting the body plan homology between cnidarians and bilaterians, between cnidarians and sponges. But now what we are trying to get to is to understand how those transcription factors and those signaling pathways are arranged in gene regulatory networks. And we have done quite a bit of antibody development and method development, and you should stay tuned for that in the next couple of years, I hope. All right. Now, so it appears that we can learn about potential homologies, so potential stages of emergence of complex animals. And this is something that we will be actively exploring for the next couple of years, I guess. But what about sponge regeneration? Well, as I said before, sponges have pretty amazing ability to regenerate. If you just slice them, they reorganize themselves, move the cells, transdifferentiate, and in the end, within a couple of days, they rebuild functional sponges. So this, starting from this mid-body slice, you have the basal part here and the apical part here, and you have the new skeleton formed and everything is fine. I really wish I could just be sliced and made into a couple of more of me so I could do my work and take care of the kids at the same time, but I guess that is now impossible. However, what is possible is trying to understand what genes are playing roles in regeneration of sponges. And the reason why it's interesting is kind of twofold. One side of that is that we know which genes are important in regeneration of other animals. So if we are comparing regeneration mechanisms, molecular mechanisms of regeneration in sponges to those that we know from other animals, we can get a better feel of whether those mechanisms are conserved and they have been preserved in some lineages of animals more than in others, or if they are independently evolved and perhaps unique to some specificity of sponge body plan or cell differentiation mechanisms. And so that is the fundamental aspect that I must say I'm most keen on. And another one is that if sponges can regenerate and we can't so much, is there something we can learn from sponges in terms of potential medical impact? And I will not be talking about that because that is something I generally think as a very futuristic kind of work, but that's something to keep in mind, that we might be able to learn something from sponges. All right, so the first fundamental question that we had was, are organizer genes, so remember genes involved in specification of the organizer capacity of the osculum, are they involved during wound healing and regeneration of sponges? And the reason why we asked that was because if you cut a cnidarian polyp, hydra, or sea anemone, you get dramatic upregulation of those genes involved in of those genes that are normally present only in the mouth of the polyp, in the organizer region, and then those genes play another role during the wound healing before they disappear in the parts, in regenerated parts that are not forming the mouth. So we thought of looking from this perspective as looking for some similarities between and potential homologies between sponges and cnidarians again. Now, this is quite a long story, and I'll show you how to access the whole story in a moment. But what I wanted to very quickly show you now is a PCA analysis that has been done using a new pipeline that somebody developed. It's called Combat-seq, and my student tonight have found, which allowed us to combine data sets from two very different experiments. And all I want you to take from this slide is that if you look at the samples that are from the intact top, so the samples containing the osculum area, and you look at the samples that are coming from the earliest stages of regeneration. And now I'm going to tell you that 3 hours is an early stage regeneration. I'm going to change what I say in a moment. So if you look at those samples taken from the earliest stages of regeneration, we had a look at 3 hours. They are on the PC2, principal component 2. They are coming together. They are close to each other. And you can see that in a way, as the regeneration progresses, it kind of, the samples move. So this is 3 hours, and this is 6 hours, and then 12 and 24, and they kind of come back together to the intact set. And we have the set of genes that is more similar to the middle part rather than the top. So this is a very global, very broad view. So we wanted to know what genes are there and if they really kind of make sense to us. And we were very excited when we have compared the genes that are upregulated at 3 hours to the list of genes that are present in the top or much higher expressed at the top of the sponge than in the middle. And we have found that there isn't many genes that are overlapping, or 77 out of 150 or 160 here, but all those genes, or many of those genes that we are finding, of course not all, many of those genes that we are finding are the genes that we are very excited to find, which are genes that are parts of the Wnt and TGF-beta pathway. So those genes, as I told you, that are part of this organizer capability. And you can see it in a bit different way here, which is the regeneration data set and the intact sponge data set, which are just slices going from top to the bottom. And you can see that all those genes are very strongly expressed in the top and not so much anywhere else. Now, this was the looking at numbers, but I'd like to show you a bit more in situ hybridizations. So this is an intact sponge with the oscular expression of a gene that we call TGF-beta U. And the U means we have found a lot of TGF-beta ligands. If you first fix a sponge and then cut it into slices, you will, as expected, not see an expression within those slices. But if you cut it and then wait 3 hours and then fix it, you will see expression of TGF-beta U at both cut areas. So here it was only around the osculum. And if you look at the higher magnification, you will see that expression of this gene is limited to pinacocytes that we know are actively migrating to form the regeneration membrane. And this is quite exciting because this pathway is also known to be involved in cell migration in eumetazoans. Now, if you look at another gene from the same pathway, which is the transcription factor, this is an intact specimen, and this is a specimen that has been cut, and then we waited for 3 hours and it was fixed. And you can see this very broad activation of this gene, and you can see something that was slightly surprising to us, which is that activation of those genes is much broader than just limited to the cut. So we have just put the paper describing these experiments on bioarchives, and I encourage you to have a look. We are also in process of submitting it to a journal. But what I wanted to say is that we've been quite happy with the fact that we found those genes expressed during wound healing and regeneration that we expected. It all puts very nicely the idea of homology together between sponges and cnidarians and the conservation of the regeneration mechanisms and so on. But what I found most stunning and totally unexpected was that at 3 hours, we had a huge difference in gene expression. And maybe I shouldn't have been so surprised. So we called it fast transcriptional activation. But then when we started working when we moved to Australia, we started working on second Capricorn. And one of the first projects that my student tonight started was to probe how fast really those changes are occurring. So we knew that by 3 hours there is a lot of genes that have changed expression, and we knew the change, the level of change is huge. It's over 1,000 times in a couple of cases of interesting genes. So we wanted to know how fast gene expression can change. So because it is for a genomics crowd, I'm going to tell you a bit about the genomes again. We have, or maybe I should start with the model system. So we are working on sponge called second Capricorn. It's been described from the Great Barrier Reef, but we are sampling it in Jervis Bay, and I know this is a bit confusing, and I'm happy to talk about it a bit later. We have started by making an in-house assembly based on Oxford nanopore and polishing with Illumina per entries, and we've got the genome size almost twice as large in the second ciliatum. The N50 wasn't very wonderful. And then we went to Dovetail Genomics. They gave us a bit of a discount with the Cole grant, and they have assembled a much more continuous genome that has the N50 of 1,000 times more of our previous one. And the genome is a bit larger, and the genome assembly is a bit larger, and we are in the process of exploring why that is. So I don't have many answers on that, so I will not talk about it, but I just wanted to let you know where we are standing with those genomes. All right. But we didn't start this project to look at genomes. Sorry to say that. Well, we are also interested in those as well. We wanted to do it so we have a model to study both homology of the body plans, cell type specification, and so on, and regeneration. So one thing that Tunay did to get started was to simply look at what is happening when you not only cut sponge into slices, but also when you dissociate cells. And those of you who have been following literature from the, say, 1905 would know that if you dissociate a sponge, the cells come back together and they rebuild the body, and in many species, they build the body completely. So those cells, so this is a little time-lapse movie, I will not show you the whole thing. The cells, when you dissociate them, they do not look upset, they look very excited, and they start crawling together, and whenever they meet another cell, they come back and they start forming those aggregates. And we know that many sponges, including to a certain extent second Capricorn, are capable to building new bodies from those aggregates, and we call it the regeneration system. So after approximately 24 hours of after dissociation, you have those nice, smooth looking aggregates, which then proceed to becoming hollow and forming spicules and so on. We have a bit of a problem of making them to become perfect sponges, but that's a separate story. What is really happening clearly is that those cells reaggregate and start building new bodies. Now, that is something that humans certainly cannot do. All right, so knowing all of that, what did we do? We spent some time discussing how many time points we are going to look at, and we ended up with a large number of time points and looking at both regeneration and reaggregation at the same time. So we would be able to compare the data sets directly, and we also used a number of replicates and each replicate was a pool of free sponges. And we still talk about this experiment in the lab because not only on the science level, but also because it contained so many different time points and so many sponges. In the end, the whole lab was together and we were slicing and passing the slices to one another, and it was quite a fun event in the lab, even though it was perhaps a bit stressful. All right, so that is the experimental design. There is a lot of data there, and I'm just going to show you very quickly what were the major findings of looking in the very first steps. So we have focused on those very first time points. We have time point 0 and time point 5 minutes, and again, we can discuss it later. We ended up using the 5 minutes as our control time point because of a variety of reasons. So what you will be seeing next will be comparisons between 5 minutes after dissociation and 15 minutes, or 5 minutes and 30 minutes. So we are talking about really short time points. And here you are looking at samples coming from dissociation and regeneration, and again, dissociation and regeneration. And this is 15 minutes, and this is 30 minutes. And the numbers are the genes or transcripts, let's call them genes, that have been found to be differentially expressed. The numbers are not as exciting as the kinds of genes that we are finding there. So overwhelming majority of those genes we are finding are transcription factors among those genes that are annotated, so we know what they are. And that means that this early response genes is upregulation of a number of transcription factors that are then going on to move on, and regulate expression of other genes. So that is very satisfying. Another set of genes that is appearing are in a bit later stages, so at 30 minutes rather than at 15 minutes, are those genes that are involved in the Wnt and TGF-beta pathway. And again, you remember those from our ciliatum study. These are genes that are early, changing their expression quite early, and this is consistent with what is happening in other animals. And now I just wanted to tell you that one of the things that is very exciting is the fact that we have Jun and Phos here, which are transcription factors that are involved in the early wound healing response in everybody, including humans. And so we have this very strong, very strong conservation of the mechanisms. All right, so I have two tiny stories to show you now. One is we wanted to know what is really regulating this very fast gene expression change. And we know that that a lot of gene expression changes, as I guess everybody in this field knows, are regulated by histone modifications. So for those of you who do not know, this is histone H3, and these are conserved across animal kingdom, including sponges, positions including lysines and arginines and so on. And there's a conserved set of modifications, including methylations and acetylations. And we generally talk about those as, for example, histone H3, K4 methylation 3, talking about the position of those amino acids. This is very obvious for a lot of people working in the field. It wasn't that obvious to me from the beginning. So Deepan, a master student in my lab, started working with Junet, who was still a PhD student and is now a senior PhD student in my lab. And the idea was that they will look at the loci that we know include genes that have dramatic expression regulation, dramatic change in expression upon dissociation or sectioning, and we look at selected set of histone modifications that are known from other animals to be markers, for example, for active promoters or repressed promoters, or other markers for active promoters or promoters and enhancers. And we'll try to see if there is correlation, and then in the next steps, maybe causality between those modifications and changes in gene expression. So we didn't go that crazy. We didn't go into all those time points. We have only used regeneration rather than reaggregation in this data set, and we only used 2 time points, 5 minutes and 60 minutes. But of course, we had free pools and biological replicates. And we have selected the 5 minutes and 60 minutes time points because at 5 minutes, as I told you before, there are practical reasons to use 5 rather than 0. We don't see any gene expression changes between 0 and 5, anything that we can notice. And at 60, we have a large number of genes that are already that have already changed their expression, including some that have actually gone through a massive upregulation at 15 minutes, and by 60, they are almost done. So there is a lot of dynamic changes of gene expression at that point. And then what he did was he took those 4 modifications, 2 controls, and ran a number of analyses. It was quite a heroic process, I would say. All right, so this is a way we look at it when we are doing the first look at those genes. We are mapping the reads along the gene bodies, and we are interested in the area between transcription start site and transcription termination site, and we are looking approximately 2,000 base pairs up and down, and this is looking at all genes we have annotated in that genome that they looked. And yes, you see it right. We think there is approximately 60,000 genes in a sponge, and we can then discuss why is that, how does that make sense. You will see those changes are not very dramatic. The difference between 0.1 and 0.25 is really pretty kind of depressing. And you can see this color coding of those different modifications. And you can see for each of them, we have the 5 minutes and 60 minutes. So there isn't any difference here. This is the control, histone H3. This is the repressed promoters. These are the active promoters. And there is some difference in those 2 acetylation marks, but it's not very satisfying. Now, if we look at the genes that have low and stable expression, so this is selected approximately 4,000 genes that just don't do much, those numbers are even more pathetic. There isn't really, you really can't see any differences here, and you see some minor differences here maybe. But now, if we look at genes with high expression, we can see that the profiles of those modifications look a bit more like we expect from the literature. And we also start seeing some differences, even though, as I said, these are genes that are supposed to have stable expression. But I will go into a bit more details here on this one. So this is a selection of 452 genes that are upregulated within 60 minutes of dissection. And here you can see that there is a really big difference between K4 methylation free and both of those acetylation marks if you compare it to the control and the K27 methylation free, which, as you remember, is repressed promoters. What we see is increase at 60 minutes for K4 methylation free and quite large increases for the 2 acetylations. What I was actually most excited about was to find that when we are looking at the small subset of genes that are downregulated, we are also able to see increase. For the first time, we see increase here in the presence of a marker for repressed promoters. So to cut it this rather long story very short, I will just give you the summary of that, which is we are pretty sure there is correlation between the histone modifications that are known to be regulating gene expression in other animals. So this is very highly conserved. Now, we clearly say correlation because we do not know much about causality yet, And to look at causality, we are going to be looking at more time points and trying to understand what is actually happening first. Is it change of gene expression or is it change in the histone marks? All right. And now, just for dessert, I'm not going to give you much data. I'm just going to tell you about what we've been doing. And I'm going to show you a number of very pretty pictures. And as you will be looking at those pictures, you can think of the questions that you might have for later. All right, so probably everybody here knows that animals have strong relationships with bacteria. At the early stages of evolution, bacteria were used as food. This is true for carnoflagellates and sponges. And we all know about the whole microbiome stuff and our skin microbiome and gut microbiome and how very important it is. And we like to think of bacteria as partners and not as pathogens all the time. Now, sponges are again in this pretty cool position where bacteria are both food and partners. There is a lot of information about roles of symbiotic bacteria throughout literature for all animals that were studied, including some about sponges. But what we really wanted to know was to zoom in on the calcareous sponges, on the models we are studying, and try to understand if there is a stable microbiome, what is the mode of transmission, and most importantly, what is the function of that. So Roger Jan was an honor student in my lab. He's now a research assistant in my lab, and he did a study that utilized a very broad range of calcareous sponges from Europe and Australia. And he did something that is, you can think of a very standard amplicon barcoding project with the standard time to pipeline. But what he found was something that got us very excited, which was that the microbiomes in different species of sponges, we can talk about those species a bit later, if those different species of sponges are very specific and they are also very stable. So for example, this is allowing you to look in a bit more detail into a couple of taxa found in Second Capricorn from different environments. So Great Barrier Reef and the temperate kelp forests. And you can see that after 3 months of keeping those sponges in the tank, those bacteria are still there, even though they have a tiny bit of contamination or food coming from the tank water. So here are the pretty pictures that I have promised, which is, if you look, we are using fluorescence in situ hybridization, and we are looking at revealing localization of bacteria that are unique to Second Capricorn. So we have cloned, we have generated six probes to reveal localization of bacteria not known from anything else, including other sponges. So this is eubacterial probe, meaning that we are looking at all bacteria. It's a section, and blue is DNA and red is bacteria. And you can see there are very different bacteria, very different shapes. Some of them are present in the larvae. We do not see this we do not see it in the negative control. And now I'll just briefly go through those different probes, so you can appreciate the different shapes and sizes of those bacteria, and the fact that in many cases we are finding them also in the larvae. And you can see we are calling them contigs, and we are just we can just get them down to the taxon, but we have no idea what they really are. So these are particularly long and very pretty. And you can see most of them we are finding also in the larvae, except of this one that has a very characteristic shape and is never found in the larvae. So what do we know? The microbial composition is very unique, very complex. Mode of transmission is often vertical. And we are now working with Chris and Annie to understand what are those bacteria might be doing. All right, and with that, I just wanted to give you the brief summary of what we are able to find if we look into sponges. So we can try to use the kind of data I was showing you into understanding the features of the last common metazoan ancestor, because that is really where I came from. And we think that we know that it had a very complex system of developmental regulatory genes. We think that it patterned the body in a very similar way to us. So the Wnt and TGF-beta pathways for the access and transcription factors like brachyury and GATA from the endomesoderm. We also know that the wound healing and regeneration mechanisms are extremely conserved. So it was very much like us. And now we also know that the way of regulation of genes was very similar to ours. And in the end, we know it had intimate and stable relationships with microbes, although obviously we don't know yet what are those functions. And I'm just going to leave you with a link here, which is a link to resources that we have produced for a number of marine models, and you are welcome to have a look there and use those for your teaching purposes if you are keen on that. All right, and I think I did a lot of talking, and I hope you guys are still with me and are keen.
Awesome. I'm doing the clapping. You should think of this as thunderous applause from thousands of people around New Zealand.
Thank you. It was a pleasure. And email me if you would like some details on anything. We have more details, and they are on topic.
Excellent. All right. Thank you very much.
All right.